Hi, my name is Gabriel Tiago Galdino, and my project is normal mode analysis applied to GPCR. G-protein coupled receptors are membrane receptors, and they are very important for drug discovery. They have 7 transmembrane domains connected by loops, and a C-terminal that is intracellular and an N-terminal that is extracellular, and they are the largest known family of membrane receptors, and around 30% of all drugs approved by FDA has a GPCR as a target. And many of the non-GPCRs, we still don't know which pathways they are involved in and which ligands bind them. So as soon as we discover these new ligands and the pathways that they are involved, new GPCRs can become drug targets. And most of these approved drugs has undesirable side effects like dependency and loss of effectivity. And GPCRs are very well studied. So 126 unique receptor complexes have structure available in PDB. G-protein coupled receptors have this name because they can bind the G-protein. So the G-protein has 3 main domains. There is the alpha domain, the beta, and gamma. And it combines a GTP molecule. So in the cell, the receptor will be in the inactive form mostly. And then after the binding of a ligand, it can change its conformations to an active state, and the active G-protein can bind to it. After the G-protein binds to it, it changes its GDP for a GTP, and this will cause a decouple of the beta-gamma subunity from the alpha subunity, and this will lead to a signal cascade. In our project, we are much more interested in the first step of the activation process, when the ligand binds to the receptor, causing a conformational change that can be represented by movements of large domains of the protein, and this will lead to a cellular response. So the ligands that can bind the GPCR can be classified in different types. So if we monitor the changes in the response according to the drug concentration, we see that the molecules that can reach a maximum activity are called full agonists. The ligands that can reach partial activation, they are called partial agonists. And the ones that don't change the basal activity are neutral antagonists. And the ones that reduces the basal activity are the inverse agonists. So to understand what an agonist does, we need to look at the population of receptors in the membrane cell. And we see that before the binding of the ligand, the inactive state has the largest population. We still have a small few proteins that are in the active state, and they are responsible for the basal activity. But after the binding of the agonist, the active state has a stabilization in its energy, and then we see a populational shift toward the active state. And after the binding of the G-protein, we have a major stabilization of the active state and a very large conformational shift. The conformational changes caused by the binding of a ligand can be detected by some molecular dynamics experiments, and they see the protein changing its conformations because the binding of an antagonist, for example, from the active to the inactive state. But these kind of experiments are very computational costly, and we are limited to a small number of molecules. An alternative for these methods could be applying normal mode analysis, that is coarse-grained, to study the dynamics of the GPCRs in complex with ligands. In normal mode analysis, what we do is to represent our protein as an elastic model, and each spring in this elastic model would be an amino acid of the protein. And the springs that connect these masses are the intra-amino acid forces that connect these masses. And from this elastic model, we can calculate the normal modes. The normal modes are movements of same frequency and same amplitude that can describe every single conformation that our system can assume. And from this, the intensity of each of these normal modes, we can detect which are the parts of the protein that has a larger chance of moving. So it could be a measure of flexibility. Our hypothesis for this project is that if we have the structure of the receptor and we dock different ligands that we know experimentally the Emax of a certain response, we can actually use these docking poses to calculate the normal modes using a normal mode analysis potential that is ANCA, that is atom type sensitive, so different ligands will have different effects into the receptor, and take into account the ligand as a node in our analytical model to calculate the dynamical signature of each of these complexes and see if we can use these dynamical signatures that are a vector of B factors that represent the flexibility of the protein in each residue to train a machine learning model to predict this Emax that we already know. For our first experiment, we decided to work with opioid receptors, and opioid receptors are very common in common drug targets in the market, and they have many drugs that are already approved that act in the receptor, and we know many experimental data about them. So we decided to work with two different opioid receptors, the mu opioid receptor, and the kappa opioid receptor. We selected high resolution structures for both of them. And for the mu opioid receptor, we selected 170 ligands with no values for Emax for GTP gamma S binding assay. And for kappa, we selected 68 ligands with no values of Emax for GTP gamma S binding assay from the Chambo database. And we removed every single small molecules and ester chains that were present in both structures and reconstructed the loops for kappa, the missing loops for kappa. The GTP gamma S binding assay is an experiment where we changes the GTP for a radioactive GTP that cannot be hydrolyzed, and we measure the accumulation of the active G protein after the treatment of a certain concentration of the ligand. After selecting the structures and the ligands, we performed 50 docking simulations for each ligand, and we selected 10 poses with the best score per molecule. And we then used these 10 poses to calculate the dynamical signature of the complex, and we built a data frame with all these beta factors and the docking scores, and we used each one of these beta factor as an independent variable in a multilinear regression model. And for the multilinear regression model, we decided to use lasso. In lasso, we want to predict a y variable, that is in our case the Emax, from a number of predictors, in our case the B factors, and find beta factors that better predict this y variable by multiplying it by each one of the B factors. And what we want to minimize in lasso is the error that makes it the error in comparison for the real prediction, so the real Emax. And also we have a lambda factor that multiplies the absolute value of the beta for each of the beta coefficients. And what we want to do is to minimize the variation of beta. And lasso has a property as when we increase the lambda factor, we reduce the variables that are less important to the prediction of the Emax to 0. And when we reduce this to 0, we have a better view of each part of the proteins are more important for the activation of the receptor or to predict our Emax. And to test our model, we decided to use two validations tests. The first one is the leave one out. In this test, we remove one ligand from the data set. We train our model to calculate the beta factors for all the rest of the data set, and then we try to predict the Emax for the 10 poses of that ligand. And we repeat this for each one of the ligands and plot to see if we have a good prediction. And this approach is very hard because we are presenting to the system a molecule that it never seen before. And it will represent better a situation where we try to predict the Emax for a new drug. So, and for both receptors, we get a R-square of 0.5 for more, and for core, we got R-square of 0.59. A second validation test that we decide to apply to our model is the 80% training and the 20% testing validation. This is an easy test, and in this test, we selected from all available poses 80% of it as a training test, and making sure that at least one pose per ligand is present in the training set. And then we use the remaining 20% of the poses to use as a test set using the beta factors calculated to the 80% ones. And then we plot to see if we get a good correlation. And for both cases, we got a Pearson's errors of 0.68. And this approach represents a situation where we have a data set that is diverse enough that each new ligand that I present to the system is a small variation, has a dynamical signature that is a small variation of the ones that we use it to train the model. Other advantage of using lasso is that first, it will reduce the coefficients that has less importance to predict the Emax to 0, and then we can map it back to the protein and see which parts of the proteins that by becoming more rigid, in the case the blue ones, or more flexible, leads to a higher Emax. And we see that for both receptors, these regions are very different. For kappa, we see that most of the coefficients with large values are centered around the binding pocket, while in more, we have that they are concentrated around the helix 6 and 5. And when we look at these positions in the literature to see if we can find some mutations that are happening in these positions with large coefficient values, we see that some of them are known to affect the activation of the receptor without affecting the binding. So this goes in agreement that we are detecting regions of the protein that can have an effect in the binding or in the G protein binding or the Emax of the response caused by the GPCR. So as a conclusion, we see that ANCHOM can use it to calculate this dynamical signature for ligands that has experimental values, known experimental values with a good precision. Even in the hard test, we get a Pearson's error that is larger than 0.5. And if we see the results for the 8 and 20 test, we see that as much as we diversify our data set by getting more experimental data of different ligands, we can significantly improve our prediction. And when we map back the lasso coefficients, we can identify these parts of the structure that has a major influence in the activation. And since ANCHOM is coarse-grained, we can easily apply this for many ligands in a high-throughput way manner. And the existence of these mutations that we see in the position that has largest values of beta factors, we see that they actually are correlated to the activation of the receptor without affecting the binding of the agonist. So they are especially interested for the dynamics of the receptor. Thank you very much.